So good afternoon, everyone. My name is Nancy Decky, and I'm a product manager at Atlas Antibodies. I would like to welcome you to this webinar, where I have the pleasure of introducing Professor Rafael Franco from the University of Barcelona. And he's going to talk to you today about his research and how he has applied MolBoolean, our new in situ proximity technology for studying protein-protein interactions. So the title of his talk is GPCR receptors, breakthrough advancements using the MolBoolean technology. So Professor Franco is a full professor of biochemistry and molecular biology at the University of Barcelona. He's leading the molecular and neurobiology laboratory. His lab is also connected to the CyberNet, which is a network center for biomedical research across Spain. Professor Franco is internationally recognized for his pioneering research on the functional relevance of the heteromerization of G-protein coupled receptors, and he has made significant contributions to neuropharmacology and neurodegeneration. Professor Franco is ranked in the top 2% of scientists globally for holding prestigious positions, published extensively, and secured multiple patents. His work spans over studying molecular mechanisms, therapeutic targets, and comprehensive collaborations, advancing the GPCR-related biomedical research. And with me also I have Dr. Mikael Malmqvist, who is the principal scientist here at Atlas Antibodies, and he's our technical expert for the MolBoolean. Dr. Malmqvist has, together with Professor Ola Söderberg from Uppsala University, developed the MolBoolean technology here at Atlas Antibodies. And he will answer any questions you might have regarding the MolBoolean technology and the MolBoolean technique. So speaking of questions, if you have any questions during the presentation, please type them in the Q&A sections, and we will address them at the end of the presentation. So please, Professor Franco, word is yours.
Thank you very much for the kind presentation. I wonder whether I'm sharing the screen is used. Are you seeing my slides?
Yes.
Okay. Thank you very much, Nancy, for the kind presentation. In this slide, I will give you a short account of the milestones on the GPCR interaction research field. This started in 1983 by indirect evidence. Luigi Agnati and Schellfuchse, let me just minimize this, emitted the hypothesis. They have been working together, and following the hypothesis, we have collaborated. My lab have collaborated with Schellfuchse and Luigi Agnati for many years, starting in late 19s. In the first reported homodimers, to my knowledge, were in 1994-95. In the lab of O'Dowd and George in Canada, that indirectly have evidence for dopamine receptor dimers, homodimers. And in my lab, also we have, in those years, evidence of adenosine A1 receptor homodimers. The most interesting part in my mind is the heteromeric, let's say the heteromers formed by two different receptors that can be for the same endogenous ligand. For instance, in 2000, there is this paper reporting the interaction between delta and mu opioid receptors by the lab of Lasmi Devi. And also the same year, we reported the first example of heteromer formed by receptors for two different neuromodulators that were the complex formed by adenosine A1 and dopamine D1 receptors. Adenosine dopamine receptors are very important in the motor control area of the brain, and also in the reward circuits of the brain. If I can go to the, okay. The heteromers provide functional diversity because these are just two examples. The dopamine D1 receptor coupled to GS, and then when you activated it, there are increases in cyclic AMP, and there is cyclic AMP signaling. When you activate the D2 instead, there is a GI coupling, so there are decreases of cyclic AMP. But when you find D1, D2 heteromers, the coupling is to GQ, and you find increases in cytosolic calcium. This is quite important because since the discovery of those heteromers, by the way, by the lab of Susan George in Canada, nobody had any clue of how to link dopamine neurotransmission to calcium signaling. That is very important for neurons. But it turns out that this heteromer provides the key answer. So this is the mediator of, or at least one of the mediators of the dopamine link to calcium signaling, which is so important in neurons. Another completely different example is the A1, A2A adenosine receptor heteromer, because this is a concentration sensor. So at low adenosine concentration, only the A1 in the heteromer is activated, and you have GI-mediated signaling. But at high concentrations, the heteromer changes the conformation, and because the A2A is activated, and then this shuts down the A1 coupling, and this only is the GS coupling mediated by A2A. So in summary, at low concentration, there is decreases in cyclic AMP. At high concentration, there are increases, and this is afforded by the heteromer. Notice that you have only one of those, you get either GI or GS coupling. But when with this heteromer, you may have reciprocal or quite diverse signaling outputs, and this explains why at some concentrations adenosine activates the release of neurotransmitter, and at some concentration does the opposite. This is because, or at least in some cases, this is because of the expression in neurons of A1, A2A receptor heteromers in the presynaptic portion of the neuron. The therapeutical, these just were samples. There is a huge diversity of, there's a lot of different possibilities that opens the field, opens the possibility to expand the therapeutic potential of targeting GPCRs. Usually, GPCRs are targeted individually, and in our mind, in the field, and for Shel Fuchsia and Luigi Agnati and for Susan George, etc., we have clear that the pharmaceutical, the drug development should take into account the heteromers. Why? Because there are expected fewer side effects. Because if you only target heteromers, you don't target all the receptors everywhere. You just target the heteromer. The question is whether it's possible to target just the heteromer, and the answer is yes. We have, and we and others have provided evidence that the conformational changes in the heteromer made possible the development of drugs that are selective for heteromers. So if you have a drug that is selective for heteromers, you are only targeting those heteromers, and this decreases the side effects and increases the therapeutic potential. This is something that we don't understand in the field, why has been continually overlooked this possibility by pharmaceutical companies. It's true that we needed tools to understand better the heteromeric, the consequences of the heteromerization, and also the other aspects of the life, let's say, of the heteromers. There were and there are challenges, and these are examples of challenges in research related to GPCRs, focus on heteromers, which is the proportion of monomers versus heteromers inside the cell or at the cell surface. This is the question all reviewers asked you, and until recently we couldn't answer. So, okay, you have heteromer, but how many? Are there a lot in comparing with the total amount of the receptors? Where in the cell are the heteromers formed? Are traveling together to the cell surface and they form at the cell surface, or they are already preformed inside the cell and they travel together to the cell surface? Also important is to know whether agonists and antagonists affect the proportion of heteromers. Where and how in the cell are those disrupted or degraded and eventually are recycled individually to form heteromers with other receptors, perhaps? Are heteromers expressed in pathological situation? How many? Because to be targets for disease, they have to be expressed. If they are not expressed, they are not cannot be targets. And the other way around, if the heteromer is expressed in the pathological situation, what you are using right now, targeting one receptor, is targeting not only this receptor, but also targeting heteromers containing the receptor. And which is the proportion of the different heteromers of a given GPCR therapeutic target? Because for a given receptor, you may have the receptor forming different heteromers with different receptors in a given cell or in a given pathological situation. We are lucky because of recent developments. Well, PLA is not so recent, but anyway, the imaging techniques have come to help us. And first appeared the PLA, the proximity in situ proximity ligation assay, that allows you to detect heteromers in cell tissues without giving this is very important. You can see the heteromers and you can convince the reviewer that the heteromer is there, but you don't know if there are a lot of heteromers or in comparison with the individual receptors. And more recently, we have the MolBoolean, that is an imaging technique. And in this case, this technique has the possibility of give you the proportion of monomers versus dimers. Let's say if the heteromer is AB, you can estimate how much of A is interacting with B of B with A, and also how the proportion of AB. So you have three different numbers that account for as I mentioned, A not interacting, B not interacting, and AB. And this is really important. This is a huge advancement for us, for the people that we investigate in this field. Now I give the word to Nancy, that will tell you how the technique works.
Thank you. So yeah, we'll briefly go through the molecular steps for the MolBoolean assay. So in this illustration, we have an interaction between protein A and B in the left panel. After binding their respective target proteins, the primary antibodies are recognized by two species-specific proximity probes. So each is conjugated with an information-containing DNA oligonucleotide, and they are called arms. And this is all in step 1. In step 2, the probe arms then hybridize to a preformed DNA circle oligo, creating a double-stranded DNA that contains two nickase recognition sequences, and they are indicated here by the arrowheads. This will be cut during the DNA nicking step in step 3. Next, in step 4, two short identifier tag oligos get incorporated into the DNA circle, followed by a DNA ligation step in step 5. The DNA circle now contains information from the two proximity probe arms. So in step 6, rolling circle amplification, RCA, creates long concatenameric rolling circle products, RCPs. The RCPs consist of the complementary sequence to the circled DNA template that is repeated thousands of times. In the last 7th step, 2 separate fluorophore-labeled tag-specific detection reporter oligos hybridize to the RCPs, revealing the identities of the incorporated identifier tags, thereby indicating the presence of interacting proteins A and B in the sample. So detection of RCPs in both fluorescent channels will be indicative of interaction between proteins A and B in the sample. So free and non-interacting proteins A and B will be detected individually, as you see in the middle and in the right panel. At these locations, only one probe arm will hybridize to the DNA circle. Only one nicking site will be revealed, allowing only one identifier tag to become incorporated into the DNA circle. And this creates RCPs where only one of the 2 fluorescent detection reporter oligos can hybridize. And these RCPs will emit light in only one single fluorescent channel. So please, Professor Franco, you can proceed.
Okay, thank you. So we use this technique for technical and for our interest in adenosine dopamine interactions, and also for technical reasons that I think are not needed to mention. We started using MolBoolean with this couple, the adenosine A2A and the dopamine D2. And so we used the ad hoc antibodies, and we performed the MolBoolean with the reagents that MolBoolean sold us. And this is, first of all, a comparison of what you see with PLA compared with MolBoolean. These are not the real colors. In white, we just find in both images, what you find in white are the heteromers. So in PLA, you only see white. And with MolBoolean, you see the 3 colors, the white that reflect the heteromers, but also you see the other 2 colors that would reflect individual dopamine or individual A2A. This is not completely true in the sense that we don't know whether these D2 or A2A are individual or not, but anyway, they are not together. So in white, they are together, and in those other colors, you find the proportion of A or A2A that are not with the D2, or the D2 that is not with A2A. This is something that was not, for us, what not, I mean, we had to work quite a bit for doing the analysis of the images taken by the confocal. And because with PLA, there is a ad hoc software, and it's relatively easy to use. To know the proportion, you have to be careful. And then, first of all, you have to do the convolution process. And also, we were lucky because we are Dr. Rafael Rivas and Esteban in the lab, and he is very good, not only in taking the photographs, but also is very good in doing different things for data analysis, image data analysis. And then he did a specific pipeline for our data using the cell profiler. And in an heterologous system, you see here the 3 possibilities: the color for A2A, the color for the D2, and in the right, you have in this case, is yellow when they are together. Green is alone the D2, and red is alone, let's say alone, but maybe alone, but in any case, in green, the D2 is not interacting with the A2A, and in red, the A2A is not interacting with the D2. And then we can now, we have to be careful, but we are confident in our pipeline to obtain results. And in this specific case, in this heterologous system, we found that the majority of the receptors are forming heteromers. So you see the yellow is much higher. The proportion of heteromers is much higher than the proportion of individual receptors. We then move within a collaboration that is working for more than 10 years with José Luis Lanciego, Alberto Rico in Pamplona, Universidad de Navarra, CIMA, and with José Luis Lavandeira García in Universidad de Santiago de Compostela, CIMUS. They have models of Parkinson's disease, and we did experiments of using MolBoolean in both models. In both models, I mean in one rodent model and in this non-human primate models, using macaca fascicularis monkeys, using MPTP that is a neurotoxic. And then we took images from the striatum. Actually, I'm not showing, I have no time to show data on the rodent model. Anyway, everything is in this paper that was published 2 years ago in Neurobiology of Disease. In this case, is sections from the macaca fascicularis, and José Luis Lanciego is, I think, is one of the best in the world, maybe the best, to do tracing. So in this case, we are only seeing at neurons of the indirect basal ganglia pathway. So there are two pathways, the direct and the indirect, and this neuron that is in my this, I don't know, this color, kind of blue, this indicates that this neuron is of the indirect pathway. And then we measure the red, the green, and the yellow in the non-lesion, lesion, and lesion dyskinetic. Dyskinetic is one of the complications of the treatment with levodopa of the patients. So when you treat a lesion animal with levodopa, some of them with time develop this dyskinesia that are abnormal movements. So we did also the counting of all three labels in section from this lesion and dyskinetic animals. What we found is, so this is one of the advantages of the MolBoolean. There's something that we thought the MolBoolean you cannot obtain, is that you can be sure that the D2 in both, well, this is the conclusion not only for the data I just show you on monkeys, but for the data of the paper, the neurobiology of disease paper, is that the D2 in primary neurons and in striatal sections is mainly forming heteromers with A to A, but the reverse is not true. So there are A to A that are not interacting with D2. But the important, both aspects are really important. But perhaps the most important one is that the D2 is always or almost always with the A to A. So the dopaminergic neurotransmission in the indirect pathway is via the heteromers D2 A to A. This is something that we didn't expect. We thought that there were D2 individually expressed or forming heteromers with other receptors. No. As soon as the D2 is expressed, it forms heteromers with A to A. We have to now to investigate where are they formed. And this is something I quote from the paper. This is a novel result since a significant proportion of D2 receptors expressed individually or forming heteromer receptors would have been expected. So we were expecting D2 not interacting with the A to A, but yeah, it's like that. The D2 are always in these cells, in these neurons of the indirect pathway are interacting always or 90% with the A to A. And in terms of pathology, taking into account the results that we found and using the models and that are reported in the neurobiology of disease paper, the A to A D2 heteromer expression increases in Parkinson's disease due to increase in D2's expression. Again, when the D2 is upregulated, and then it binds immediately to the A to A. Still, there are A to A that are free, let's say free, but the D2 that is upregulated in Parkinson's disease, again, is forming heteromers with the A to A. So the A to A D2 receptor heteromers in neurons of the indirect pathway are therapeutic targets in Parkinson's disease. Levodopa in this pathway is targeting the heteromer. So there is targeting, let's say, it's not correct. I would put it in more correctly. The levodopa is targeting the D2 receptors in patients that are forming heteromers. So they're targeting D2 in heteromeric context with the A2A. And just the last is just something that is just ongoing. You have to put something different. We are looking using MolBoolean to another heteromer. In this case, again is the A2A, but now with the cannabinoid CB2. Both are upregulated when microglia cells are activated. And then we were interested in see with the proportion. We're interested, and still we are doing the analysis of data. But anyway, you see there is an increase in the expression of these microglial cells, and we are counting how many are individual or how many are forming heteromers, A2A, CB2 heteromers. This is ongoing. The idea is to try to find a way to skew microglia towards the neuroprotective phenotype. So with these results, we are sure that, well, we were sure. The hypothesis is quite, let's say, the hypothesis, the Occam's-Razor hypothesis is that these heteromers are targets for skewing the microglia. We have to find a way, which is the pharmacological way, agonists, antagonists, or one or the other, the way to skew them towards the neuroprotective phenotype. So this is what we are doing, among other things, that we are taking real advantage of the MolBoolean because this has opened a lot of possibilities. And this is all, and thank you very much for your attention.
Thank you so much, Professor Franco, for this nice presentation, sharing this very interesting data with us. Let's see if we have any questions. Have been popping up a few. First one. Given the wide focal plane and limited resolution of the confocal microscope, are you sure you are not collecting overlaid receptors instead of heteromers? Do you see similar profiles with the TIRFM? I think that's for you, Professor Franco.
Yeah, we measure different depths at different layers, so we take this into account. Yeah. So we are pretty sure of what we are. I mean, in the final data, the proportion is quite reliable.
Yes.
If I may, I could add on to that slightly. Rafael showed you that you developed your own image analysis pipeline, but also with MolBoolean, we do have a MolBoolean pipeline that you can download that comes complementary with the product, and it can be run in Cell Profiler. And as part of that pipeline, we have steps where we are segmenting the rolling circle products, and in those steps, we are shrinking the objects to smaller sizes to avoid false positive overlap, and that you can shrink those objects to defined pixel size to avoid more or less the false positive signals. I also want to mention that the pipeline includes steps where you set thresholds for the intensity for the objects that should be counted. And if you have objects that are not in the same focal plane, they will have if you have an object that's out of focus, they will have a lower intensity, and that could be then removed through the thresholding during the image analysis pipeline.
Okay. So let's see. How can A2A receptor antagonists help treat Parkinson's disease?
Well, actually, this is something that in preclinical research, the A2A specifically, just the A2A, the preclinical assays indicate that targeting the A2A is neuroprotective. As you know, I'm sure you know that it's not easy to demonstrate neuroprotection in humans. But so the regulators can only approve at this point drugs that are good for symptoms, and in this particular case, there is already one A2A antagonist that has been approved in Japan, and in the States. And I take profit to say that I think longitudinal studies would demonstrate whether this treatment with this antagonist will delay the progression of the disease or not. But at least at the moment is approved for symptomatic relief together with levodopa. And maybe I should add also that we have shown that antagonists have different affinities for the A2A in different heteromeric contexts. So this is something related to what I mentioned about the heteromer selective drugs. So depending of whether the A2A is in, let's say, presynaptic versus postsynaptic heteromeric complexes, the different antagonists behave differently because of the affinity is different.
Okay, so has MolBoolean been validated in both cells and tissue?
I think so, but maybe Mikael can add to that.
It is validated in both cells and tissue, yes.
Yes.
And then here, have you looked at GPCR receptor tyrosine kinase heterodimers?
Not in my lab yet.
What is the proximity of the dimers in nanometer scale?
They are interacting, direct interaction. We had model, and we have used transmembrane peptides that have the same transmembrane sequence that penetrate membranes. It is because are linked to a peptide. And this, when you disrupt the interaction, you can detect some functional impairment. So we have done those experiments. We have modeled, and in other labs they have done something similar, and there is a direct, the transmembranes are interacting together. Maybe I could add that in the A1, A2A, there is a tetramer with one GI and one GS. So there are two A1 receptors, two A2A receptors, and below they have one GI and one GS.
So one more for Professor Franco. How is the striatal A2A receptor regulated in Parkinson's disease?
Good question. Let's say the hypothesis right now is that it changes the composition of heteromers, but I don't think there is much change in expression. I must add for something that I could have added during the presentation, that the place where the A2A is most heavily expressed in the whole body is in the striatum. So I think that the changes is the proportion of heteromers with different GPCRs, but I don't think the A2A is changing much, at least globally. I don't know if in the specific subpopulation of neurons.
So now we have two questions for Dr. Malmqvist, I think, here. Can MolBoolean be used with any primary antibodies? And what is the volume of the kit and how many assays does it cover?
Yeah, so the MolBoolean kit is currently only for mouse and rabbit primary antibodies. But if there is an interest for any other primary antibody species, we are happy to provide you with a custom quotation to see whether we can help you to provide a secondary antibody towards another species. So just send us a request, we'll see. And for the size of the kit is The total working volume is 4.8 milliliters, which is approximately 120 assays in cells and 60 assays on a tissue slide section.
Yes. So here, do you have any experience in studying GPR18 receptor using this method? Is there a possibility to study if GPR18 creates heteromers with other receptors? For Professor Franco?
I have no experience, but yes, something that have been just said is that you need, at the moment, you need, let's say, a monoclonal and polyclonal antibodies. This is the reason. Well, we are interested in the A2A for since I was young, let's say. But, also, we have been lucky because Joel Linden developed a monoclonal antibody against the A2A maybe 3 years ago. So this is among the few receptors that have monoclonal antibodies that are really good. So maybe the issue is to find the right, define the couple, the two receptor A and B, and you need one for antibody monoclonal and one polyclonal. And again, if MolBoolean apparently is willing to develop for other species as well.
So I think the last question for Professor Franco, does interacting GPCRs see their binding site or ligand channel diffusion changing compared to monomer form?
Sorry, can
Yeah,
you?
I don't think, does interacting GPCRs see their binding site or ligand channel diffusion changing compared to the monomer form?
I don't know the answer, but we are doing right now in silico studies, trying to see something like like that, but also maybe I don't know if it's related directly. Again, I would repeat that the conformational changes affect the orthosteric center. And in the CB2, we have shown also that there are vestibule. So there is also orthosteric center, a vestibule, and then when there is an interaction of the CB2 receptor with another receptor, like the A2A, this changes. Yeah.
And the last one for Dr. Malmqvist. What is the next steps in your studies with MolBoolean?
Okay, I don't know whether that's for me or for
No, maybe it must,
Professor Franco.
maybe it can be for both.
Because I'm not doing many studies.
I would like to be younger because with MolBoolean and with also with in silico tools, there is a lot of possibilities to look for. Let's see. Let me try to put it in the right order. First of all, to do colocalization of heteromers and monomers with intracellular organelles. And this is possible. This is possible. There are so many different fluorophores that is possible. This is possible. And without MolBoolean, this type of experiment with PLA gives you some information, but not the one that, it's much better if you use MolBoolean with a marker of mitochondria or endosomes but also I forgot to mention related to a previous question of the GPR18 or yeah, this is something that I forgot to mention in real cells, not in real cells, in tissue, no, but in heterologous systems, you can obtain the plasmid. Actually, now there is a company. May I say the name of the company or not? Well, why
yeah.
not? Vector
Why
builder.
not?
Vector builder. You can buy. You can tell them what you need to express, and they give you quickly and for a good price. And then we have obtained a lot of plasmids, for instance, for receptor having the hemagglutinin tag at the N terminal. This way you can detect using monoclonal for HA, for hemagglutinin, which is monoclonal. Also, it's true that I forgot to mention that you don't need a monoclonal against the receptor A and a polyclonal against the receptor B. If they are tagged, for instance, with hemagglutinin, you can use a monoclonal against the tag. So this is something that is very attractive to use tag, but of course you need to transfect cells.
Can I add something there, Professor Rafael Franco? Also, when you do use a system where you overexpress proteins, it's very valuable to use MolBoolean, because then you could normalize your interaction data to the total target levels. So obviously you might be overexpressing your proteins of interest, and you could get false positive interactions just due to overload of protein in close areas, even though they're not interacting. So then you could normalize your interaction data then to that overexpression of the proteins as well.
Sure. And also in my mind, I would do in this situation with hemagglutinin tag or with other tag, I would combine to do solubilization of the membrane and not to see how many of each are inside the cell and how many of each are in the cell of each are on the cell surface. So with tags, you don't need to solubilize the membrane. Then you only detect what is on the cell surface. That is something quite attractive because in many situations you find the whole thing and you don't distinguish what is on the cell surface or what is inside. So this is something that is possible, and with MolBoolean it's very. I mean, you could have just on the cell surface the proportion of each.
Exciting.
Okay, there are a few that has written, thank you for a great presentation. And that I want to do again also, thank you for a very nice presentation. And if there are no further questions, we will end this webinar. And I wish everyone a nice afternoon and evening. Thank you all for calling in.